Figure 3.2. 3.3 pteridophytes. The pteridophytes include hostales and ferns. Pteridophytes are used for medicinal purposes and as soil binders. They are also frequently grown as ornamentals. Evolutionarily, they are the first terrestrial plants to possess vascular tissues, xylem and phloem. You shall study more about these tissues in Chapter 6. The pteridophytes are found in cool, damp, shady places, though some may flourish well in sandy soil conditions. You may recall that in bryophytes the dominant phase in the life cycle is the gametophytic plant body. However, in pteridophytes, the main plant body is a sporophyte which is differentiated into true root, stem and leaves. Figure 3.3. These organs possess well-differentiated vascular tissues. The leaves in pteridophyta are small microphyles as in Selaginella or large macrophylls as in ferns. The sporophytes bear sporangia that are subtended by leaf-like appendages called sporophylls. In some cases, sporophylls may form distinct compact structures called strobili or cones. Selaginella, aquasetum. The sporangia produce spores by meiosis in spore mother cells. The spores germinate to give rise to inconspicuous, small but multicellular. Free-living, mostly photosynthetic thalloid gametophytes called prothallus. These gametophytes require cool, damp, shady places to grow. Because of the specific restricted requirement and the need for water for fertilization, the spread of living pteridophytes is limited and restricted to narrow geographical regions. The gametophytes bear male and female sex organs called antheridia and archegonia, respectively. Water is required for transfer of anthrazoids, the male gametes released from the antheridia, to the mouth of archegonium. Fusion of male gamete with the egg present in the archegonium result in the formation of zygote. Zygote thereafter produces a multicellular well-differentiated sporophyte which is the dominant phase of the pteridophytes. In majority of the pteridophytes all the spores are of similar kinds. Such plants are called homosporous. Genera like Selaginella and Salvinia, which produce two kinds of spores, macro large and micro small spores, are known as heterosporous. The megaspores and microspores germinate and give rise to female and male gametophytes, respectively. The female gametophytes in these plants are retained on the parent sporophytes for variable periods. The development of the zygotes into young embryos take place within the female gametophytes. This event is a precursor to the seed habit, considered an important step in evolution. The pteridophytes are further classified into four classes: Psilopsida, Psilotum, Lycopsida, Selaginella, Lycopodium, Sphenopsida, Aquasetum, and Pteropsida, Dryopteris, Pteris, Adiantum. 3.4 Gymnosperms. The gymnosperms, gymnos, naked, sperma, seeds, are plants in which the ovules are not enclosed by any ovary wall and remain exposed, both before and after fertilization. The seeds that develop post-fertilization are not covered, i.e., are naked. Gymnosperms include medium-sized trees or tall trees and shrubs. Figure 3.4. One of the gymnosperms, the giant redwood tree sequoia, is one of the tallest tree species. The roots are generally tap roots. Roots in some genera have fungal association in the form of mycorrhizal pinus, while in some others, cycas, small specialized roots called coralloid roots, are associated with N2-fixing cyanobacteria. The stems are unbranched cycas or branched pinus, cedrus. The leaves may be simple or compound. In cycas, the pinnate leaves persist for a few years. The leaves in gymnosperms are well adapted to withstand extremes of temperature, humidity and wind. In conifers, the needle-like leaves reduce the surface area. Their thick cuticle and sunken stomata also help to reduce water loss. The gymnosperms are heterosporous. They produce haploid microspores and megaspores. The two kinds of spores are produced within sporangia that are borne on sporophylls, which are arranged spirally along an axis to form lax or compact strobili or cones. The strobili-bearing microsporophylls and microsporangia are called microsporangiate or male strobili. The microspores develop into a male gametophytic generation which is highly reduced and is confined to only a limited number of cells. This reduced gametophyte is called a pollen grain. The development of pollen grains take place within the microsporangia. The cones bearing megasporophylls with ovules or megasporangia are called macrosporangiate of female strobili. The male or female cones or strobili may be born on the same tree Pinus. However, in cycas male cones and megasporophylls are born on different trees. The megaspore mother cell is differentiated from one of the cells of the nucellus. The nucellus is protected by envelopes and the composite structure is called an ovule. The ovules are born on megasporophylls which may be clustered to form the female cones. The megaspore mother cell divides meiotically to form 4 megaspores. One of the megaspores enclosed within the megasporangium develops into a multicellular female gametophyte that bears 2 or more archegonia or female sex organs. The multicellular female gametophyte is also retained within megasporangium. Unlike bryophytes and pteridophytes, in gymnosperms the male and the female gametophytes do not have an independent free-living existence. They remain within the sporangia retained on the sporophytes. The pollen grain is released from the microsporangium. They are carried in air currents and come in contact with the opening of the ovules born on megasporophylls. The pollen tube carrying the male gametes grows towards archegonia in the ovules and discharge their contents near the mouth of the archegonia. Following fertilization, zygote develops into an embryo and the ovules into seeds. These seeds are not covered. 3.5 Angiosperms. Unlike the gymnosperms where the ovules are naked, in the angiosperms of flowering plants, the pollen grains and ovules are developed in specialized structures called flowers. In angiosperms, the seeds are enclosed by fruits. The angiosperms are an exceptionally large group of plants occurring in wide range of habitats. They range in size from tiny, almost microscopic wolfia to tall trees of eucalyptus over 100 meters. They provide us with food, fodder, fuel, medicines and several other commercially important products. They are divided into 2 classes, the dicotyledons and the monocotyledons, figure 3.5. The dicotyledons are characterized by having 2 cotyledons in their seeds while the monocotyledons have only 1. The male sex organ in a flower is the stamen. Each stamen consists of a slender filament with an anther at the tip. The anthers, following meiosis, produce pollen grains. The female sex organ in a flower is the pistil or the carpel. Pistil consists of an ovary enclosing 1 to many ovules. Within ovules are present highly reduced female gametophytes termed embryo sacs. The embryo sac formation is preceded by meiosis. Hence, each of the cells of an embryo sac is haploid. Each embryo sac has a 3-celled egg apparatus, 1 egg cell and 2 synegids, 3 antipodal cells and 2 polar nuclei. The polar nuclei eventually fuse to produce a diploid secondary nucleus. Pollen grain, after dispersal from the anthers, are carried by wind or various other agencies to the stigma of a pistil. This is termed as pollination. The pollen grains germinate on the stigma and the resulting pollen tubes grow through the tissues of stigma and style and reach the ovule. The pollen tubes enter the embryo sac where 2 male gametes are discharged. One of the male gametes fuses with the egg cell to form a zygote syngamy. The other male gamete fuses with the diploid secondary nucleus to produce the triploid primary endosperm nucleus pen. Because of the involvement of 2 fusions, this event is termed as double fertilization, an event unique to angiosperms. The zygote develops into an embryo with 1 or 2 cotyledons, and the pen develops into endosperm which provides nourishment to the developing embryo. The synegids and antipodals degenerate after fertilization. During these events the ovules develop into seeds and the ovaries develop into fruit. The life cycle of an angiosperm is shown in figure 3.6. Plant life cycles and alternation of generations. In plants, both haploid and diploid cells can divide by mitosis. This ability leads to the formation of different plant bodies, haploid and diploid. The haploid plant body produces gametes by mitosis. This plant body represents a gametophyte. Following fertilization, the zygote also divides by mitosis to produce a diploid sporophytic plant body. Haploid spores are produced by this plant body by meiosis. These in turn divide by mitosis to form a haploid plant body once again. Thus, during the life cycle of any sexually reproducing plant, there is an alternation of generations between gamete-producing haploid gametophyte and spore-producing diploid sporophyte. However, different plant groups, as well as individuals representing them, differ in the following patterns. 1. Sporophytic generation is represented only by the one-celled zygote. There are no free-living sporophytes. Meiosis in the zygote results in the formation of haploid spores. The haploid spores divide mitotically and form the gametophyte. The dominant, photosynthetic phase in such plants is the free-living gametophyte. This kind of life cycle is termed as haplontic. Many algae such as volvox, Spirogyra and some species of Chlamydomonas represent this pattern, figure 3.7a. 2. On the other extreme is the type wherein the diploid sporophyte is the dominant, photosynthetic, independent phase of the plant. The gametophytic phase is represented by the single to few-celled haploid gametophyte. This kind of life cycle is termed as diplontic. An alga, Fucus sp. represents this pattern, fig. 3.7b. In addition, all seed-bearing plants, i.e., gymnosperms and angiosperms, follow this pattern with some variations, wherein the gametophytic phase is few to multi-celled. 3. Bryophytes and pteridophytes, interestingly, exhibit an intermediate condition, haplodiplontic. Both phases are multicellular. However, they differ in their dominant phases. The dominant, independent, photosynthetic, thyloid or erect phase is represented by a haploid gametophyte, and it alternates with the short-lived multicellular sporophyte, totally or partially dependent on the gametophyte for its anchorage and nutrition. All bryophytes represent this pattern. The diploid sporophyte is represented by a dominant, independent, photosynthetic, vascular plant body. It alternates with multicellular, saprophytic, autotrophic, independent but short-lived haploid gametophyte. Such a pattern is known as haplodiplontic life cycle. All pteridophytes exhibit this pattern, figure 3.7c. Interestingly, while most algal genera are haplontic, some of them such as Ectocarpus, Polysiphonia, Celts are haplodiplontic. Fucus, an alga, is diplontic.